So it gives me great pleasure to introduce our speaker today. Dr. Melissa Davis is certainly relatively new to Morehouse School of Medicine, but is a nationally and recognized leader in genetics and genomics. She came to Morehouse School of Medicine just a few years ago, but since then has been um, um, helping us to realize a number of firsts. And certainly, she is the first Morehouse School of Medicine Georgia Research Alliance Distinguished Investigator, our first director of the Institute of Translational Genomic Medicine, and she's helped us to realize a major win related to a very large award that I trust you will share a bit about. But more importantly, her work represents an important cross-departmental, research and translational area, and that is of genomics and the importance of using Ancestry to resolve health disparities. Dr. Davis, we're so pleased that you've given us your time today and look forward to learning from you. Welcome.
Well, thank you so much. I really appreciate the invitation. Always a joy to come across the city and convene with my colleagues here at Emory. So first of all, just a disclosure that I am a cancer researcher. So most of what I'm going to talk to you about is sort of in that context of, population studies that we've done. And I'm going to try to stay on time. So I'm going to tell you a couple of stories sort of around, first of all, feasibility and the rationale as to why we started using genetic ancestry. And then I'm going to try to weave it into specifically in your area in terms of diabetes, why ancestry might be important, but then also not to completely remove the other aspects of identity. So we'll come back to that. While I show you my disclosures, I also want to take a moment to introduce you to my research partner, Dr. Lisa Newman, who's the Chief of Breast Surgery at Newark Presbyterian Hospitals. So recently, the National Academy for Sciences, Engineering, and Medicine published sort of a book, if you will, about descriptors, population descriptors. And the back of that book is a flow chart, sort of a decision tree or set of decision trees that they put forward to the community as an effort to try and, resolve some of the tension and confusion with how we study populations now that we are more aware of the diversity across the world. So particularly when we're talking about biological determinants versus social determinants, it really does matter how you stratify your work, how you determine who you're studying, and how you describe them. And so in this work, they talk about when it's appropriate to use genetics and ancestry versus other ethnicity labels. And at the end of the day, the answer to the question of what you should do is really within the context of what the question is that you're asking. So if you're asking a purely biological question and you want to understand how genetic networks are impacted across a population, then it will behoove you to measure, describe, characterize the genetic background of that population, because then you'll be fully informed about the different alleles or gene versions, that you're actually trying to study across this population. But if your question is really in the context of the social construct, so how someone's environment or their lived experience is impacting their outcome, then you probably do want to take note of who they identify as, but more importantly, the experiences that they've lived because of the construct that's around them. So the problem that we work on is the one of breast cancer mortality being 40% higher in African American women than white women in the United States. But it wasn't always the case. So if we look at sort of the historical data that shows us the mortality rates of both prostate and breast cancer, I'm going to point you here to the line at the bottom at breast cancer. Prior to 1990, it really didn't matter if you were Black or white. Everyone had the same opportunity to survive a breast cancer diagnosis. But as the disparity divided the population by race, as you can see clearly here, the white versus Black gap is very clear and poignant in the prostate categories. There was also this correlation, if you will, with socioeconomic status and social determinants. So this map that you see here is the percentage of poverty county by county across the United States. Later, you're going to see a map that shows the density of African Americans across the United States. And unfortunately, where we see marginalized populations like African Americans, you will also see the correlation of poverty. But it's not necessarily that all African Americans live in poverty, right? So then we come back to the mortality lines. Now, if we separate the race groups, not only just by Black and white, and here the blue lines represent the Black population, the purple lines represent the white population. But if we pay attention to whether the patients lived in a county that was a poverty county, so a poor community, or if they lived in affluence, so they lived in a richer county, which then should separate the population by sort of that socioeconomic status, impact on access, right, to care. Then if it was truly just that issue of access, then we should see a resolution that would have the people who live in poverty having a similar mortality line. But instead, what we see here is that Black patients who live in affluence still have a higher mortality rate than white patients who live in poverty. So that tells us that it's not an SES status issue. There's something else driving this disparity. Now, in the case of breast cancer, as I mentioned, this emerged in 1990. And what happened at that point in history, medically speaking, was first of all, the advent of hormone-targeted therapies for breast cancer. So this is when we started to treat breast cancer patients with drugs that specifically targeted estrogen and HER2. But we also have some advents in technology that allowed us to screen better and identify breast cancer at earlier stages. And some of that didn't necessarily translate into the Black community. So what happens then at that advent of these technological advances and medical advances is a stark decline in the white population in their mortality. But we see the Black population still has the same level of mortality for another decade. And then we start to see a slight decline. And so what is driving those differences? As I look at sort of the determinants of health disparities, and this is a typical outline of how we think about sort of the historical path that we've walked in terms of translational research. One of the things that we try to do is take a global perspective, because if we're talking about biological determinants, then it should transcend just the communities that we live in right now. It should be larger than Atlanta, larger than the state of Georgia, even larger than the country of the US. If this is a genetic ancestry issue, then we should be able to see it globally, right? So we term sort of this study oncologic anthropology. It's a term that Dr. Newman came up with. And we make sure that we, for instance, pay close attention to the genomic variables here. Now, when we do sort of the epidemiological assay, or not assay, I'm a wet bench biologist, so everything's an assay when it's an experiment, but when we do this type of analysis of mediating factors, so we're looking at all of the different factors that we're able to measure and then ask what is the sort of quantity or the influence of each one of those factors, we don't see the things that are associated as much with socioeconomic status having as strong an influence as, in this case, tumor characteristics. And this is specifically for breast cancer. Now, one of those tumor characteristics is hormone receptor status. And so at a population level, we actually see that Black women have a higher burden of a certain type of breast cancer. And it is called triple negative breast cancer. And by definition, it is just a diagnosis of exclusion. And all it really tells us is what's not present in the tumor. And then it excludes that patient from having the targeted endocrine therapies. But it also happens to be one of the most aggressive forms of breast cancer. We see it diagnosed as an interval cancer very often, meaning a patient might be faithfully going for mammography screening, but between screenings, she identifies a tumor. And so it grows from nothing to something that's palpable before the screening can even be beneficial. But we also know that in the Black population, African Americans are going to be twice as likely to have that diagnosis. And said another way, over 60% of triple negative cases occur in African Americans. So this is data that I'm showing you for, it's about 20 years worth of data. It includes hospital systems in Detroit. So this is Henry Ford Health Systems, which is where I was when I joined with Dr. Newman. And then the other population here is New York Presbyterian Hospital Systems in New York. So the point of this study was really to determine whether or not screening impacted survival. There's sort of this notion that African Americans don't necessarily screen as much or don't have the same quality of screening. And so we wanted to know, at least in these healthcare systems where the patients are being treated by the same doctors with the same standard of care, if screening actually made a difference. So the first thing is to show that there was no difference in screen detection between the two populations, white Americans and African Americans. There was also not a significant difference in stage. Okay, so they were all pretty much diagnosed primarily at early stages. And this is all triple negative breast cancer. So what we expect to see just by screen versus non-screen detected cancers is that the screen detected cancers, which are typically earlier stage, are going to have a better overall survival because they're diagnosed earlier, it's easier to treat and cure them, as opposed to non-screen detected cancers, which is when the patient comes into the clinic because she has, found some anomaly, with her body. So we see this persist in the white American population, but when we look at the Black population, of the African Americans, what we didn't necessarily expect to see was this widening of the gap between screen and non-screen detected outcomes. And again, these are patients who are all being treated at the same system with the same, at the hands of the same clinicians. And so the clinicians will see this data, and the first thing they'll say is, we need to get all the Black patients on the blue line. We need to screen more in the Black population because that's that's how we're going to save them. But even that blue line is a little lower, right, than the blue line in the white category. So I look at this and I think the treatment is not working as effectively in this population for the same disease, even at the hands of the same clinicians and the same system. So what are the causes for these differences? Well, when we think about outcome from cancer, similar to maybe diabetes, it's really the management of the disease that determines whether a patient, especially if it's an early catch, right? If it's an early stage, the management of the disease is really what dictates what the outcome will be. And so if we think about whether or not treatments have been effective in this population, one of the things we note is that in African Americans, there's also a higher rate of adverse events, meaning there are therapy-induced outcomes that are threats to their health. So for instance, they may not die of metastases and organ shutdown, they may die a large percentage of the time from heart disease because there's a treatment-induced cardiotoxicity that creates irreparable damage in their cardiovascular system. But why is that? Why is it so specific to the African American population? Well, if we take a step out of breast cancer and look more broadly across the cancer space, we actually have data from pharmacogenomics that tells us there are gene variants that are predictive of whether or not a person will be more susceptible to cardiotoxicity when given the very chemotherapies that triple negative patients get. And I'm showing you two of them. If we look, and what you're seeing here is the global distribution of those specific variants that cause cardiotoxicity predisposition. And if we look across the population in terms of the frequency, what we see is the highest frequency of these alleles are in populations that are of African descent. So African American, Afro-Caribbean, and Africans living on the continent. One of these even includes South Asian populations as the ones who have the highest prevalence of these alleles. And so why then aren't we screening our patients for these particular variants before they're given chemotherapy. I can tell you that it's not being done in breast cancer, but it is done in pediatric cancer. So there's a sort of concept here of sort of better management with children than we do with adults. But if we could identify incompatibility to systemic therapies prior to even or delivering these therapies, and having an African or ancestry associated disparity in the outcome could potentially prevent a large amount of the disparity that we see in mortality. But why isn't this sort of just common knowledge and something that we generate systematically across the spectrum of cancer research? Well, first of all, the data that underpins what we understand in terms of the genetic landscape in most genetic disorders, and cancer is a disease of the genes, the data is primarily derived from European ancestry participants. Okay, so what these graphs show on the left is that over 95% of the genomic data that we currently have access to is from the people of European ancestry, despite that population not being the global majority. So then said another way, less than 5% of the genomic data represents over 80% of the world. And so then that tells us we don't necessarily have a great concept then of genetic variants that are clinically relevant to any other population than the European population. And we see this translate into clinic as sort of inaccurate clinical tools, okay? So we see it replicate in prognosis algorithms, we see it propagate in genomic testing, and what I'm showing you on this graph is a mutation panel for cancer genes, or a set of different panels for cancer genes. And what this indicates is that if you're of any other ancestry than European, you're more likely to have a variant of unknown significance. And what is that? It's actually a mutation in a cancer gene, but it's one that we've never seen before, and so we don't know whether or not it's pathogenic. And anything that's unknown in the clinic space is by definition also unactionable. So typically, if you had a mutation in a cancer gene that we knew was pathogenic, meaning we know that you lose the function of the gene because of this mutation, we've shown and we have evidence that having this mutation increases the likelihood or risk of you having a malignancy, then typically another arm of the healthcare system opens up to you. So if you are a positive BRCA carrier, for instance, then you're automatically qualified for more surveillance, more screening, new technologies of screening, and even prophylactic clinical interventions to help reduce your risk. But if your variant is of unknown significance, then all you then have is the knowledge that you have a mutation in a gene that could cause cancer, but you cannot do much about it. Unless you happen to be very affluent, right? And you can have doctors that will ignore the status quo and provide those services. So now steps in this global perspective. With Dr. Newman, as a breast oncologist, surgical oncologist, before I met her, about 15 years, she spent time in both West and East Africa training general surgeons to be surgical oncologists, because typically clinicians on the continent don't have the education opportunities as a whole as those in westernized society. So she took it upon herself to go into these areas and provide those trainings. So one of the things she identified or observed, I should say, is that in West Africa, in Ghana, where she primarily was, she saw that the patients that were coming to her for surgery were much younger. And once we had access to the tissue, we found that the majority of them were triple negative, okay, about 80%. Then in Ethiopia, which is our other primary site where she does clinical training, she saw that those populations primarily have the same types of tumors as, say, the European Americans in the United States. So at that time, she was at Michigan, University of Michigan. So then when we look just on the continent at the regions across Africa, what we see is that West African nations have a higher rate of triple negative breast cancer are the highest rates than other regions on the continent. And then globally, when we look at sort of the frequency of triple negative breast cancer, the highest rates are in sub-Saharan Africa. And then in every nation where we can separate the population based on self-reported ancestry, it is the Black or African component of the population that has the highest rate of triple negative breast cancer. And so that tells us that there's a potential connection to African ancestry, particularly since we see it globally associated with this particular phenotype that could play a role in the mechanism of either cancer susceptibility or survival based on treatment compatibility, and certainly the biology of these tumors. Now, the reason why we think that this is an important topic that we need to dive into and pay closer attention to is because this population, the African diaspora, is unlike any other human population. So most of the human populations that we can appreciate across the world were derived from migration events. So founder populations that include a group of individuals, relatively small, okay, so some portion of that population that moves to another geographic region. And then what we have is just normal genetic drift, maybe some pressure from the new environment, but typically normal genetic drift that would then only account for a small change, right, between the original population and the found population. But in the case of the African diaspora, what we have is a continuous gene flow that happened over centuries. It wasn't a single event, it was millions of Africans that were displaced, specifically across the Caribbean and the Americas. And so with that gene flow comes this flow of African ancestry. And so that's why globally, because that sort of flow also brings with it the genetic background and therefore the susceptibilities to the same diseases, we can see this global imprint across the world related to triple negative breast cancer, or at least that's the theory. So we measured West African ancestry in our multi-ethnic cohort, which would include people of various backgrounds. And we found that the patients who had the highest percentage of West African ancestry were more likely to have a triple negative breast cancer diagnosis compared to all the other types of breast cancer. But we also looked more carefully just at the African American population, which has this sort of range, if you will, of West African ancestry. And even still then, we saw persistently that even in African American groups, the highest percentage of African ancestry was associated with a higher likelihood of triple negative breast cancer. But what does that even mean, right? What do I mean when I say being of a certain ancestry, or the more of it you have in your genome, the more likely you are to have a certain type of predisposition to a disease? What we're really saying here is that there is a genetic predisposition that is in the background of African ancestry. And so then the more you have of it, the more likely you are to have that particular gene variant. But because we know that ancestry in the admixed population is stochastically spread across all of the chromosomes. So we really can't just globally assess ancestry and think we're going to find the thing that we're looking for. If we applied African haplotyping, for instance, to this question, what we're really trying to find is that across each patient's chromosomes, which again can have a mixture of different ancestries, we're looking for the region that is shared across all of them that has this African background. So across the top here, what that would look like is sort of a lot of noise in terms of across the population, what is the proportion of African ancestry, or the average of African ancestry physically as you go across the chromosome. But then there'll be a spike, if you will, in the cases where African ancestry is shared across most of them, which would determine then for us where the region of where the genetic allele resides, right? And we could find this regardless of whether the patient has 10% ancestry, 70% or 80% African ancestry. We really care about this region that's harboring the allele that we're looking for. And we haven't found it yet. We have some candidates, but we haven't found it specifically yet. So we have an intentional recruitment paradigm, if you will, where we are capturing the diaspora by having targeted enrollment in areas where we know there are the densest populations of people of African descent, particularly in the United States. And we also have recruitment happening on the continent of Africa because we also need contemporary, solid, if you will, African chromosomes to hone in on the difference between random background and what is sort of the African background to help us filter which regions are important. So this is on the shoulders of a project called the Polyethnic 1000 Project, which was spearheaded by Dr. Harold Varman, as former NCI NIH director, Nobel laureate, who decided that he wanted to try and fill the gap of diversity. And so across all of these cancer centers in New York, what they decided to do was enroll 1,000 patients with various forms of cancer into this study where we would perform whole genome sequencing. And the only inclusion criteria was that they can't be white. They can't self-identify as European. They can be of any other ethnicity or ancestry. And so we have sort of this enrichment now of individuals, depending on the teams, because we have about 6 different teams as a part of the Polyethnic 1000. My team focuses on African Americans and Afro-Caribbeans and Afro-Latina populations in the borough area, in the New York metro area, but then other teams focus on Asian populations, such as the lung cancer team, or even Middle Eastern populations, such as the bladder cancer team. But all in all, these are sort of all the tools that we're using. So we're measuring gene expression, we're measuring genetic ancestry, we're looking for variants, single nucleotide variants, we're looking for structural changes that are unique to these populations that potentially hone us into what some of the predispositions are. One of the genes that we think we're on the right track with is called ANCL1. Now, ANCL1 has an allele that has been on GWAS studies, but it was found to be a protective allele. So this G variant at a certain position was shown to be protective against poor outcomes of cancer. And so it was ignored as far as research is concerned. But when we look more carefully, and again, we talked about this bias in the population of being primarily European, when we look more carefully, African Americans tended to have the other variant, which was a risk variant. So it's gone unstudied until now. What's interesting is when we look at the structure of the gene product and what changes from one variant to the other, it's clear that one of them inserts this tryptophan, and if you remember your biochemistry, tryptophan pretty much destroys structures, and so we know that there's potentially a difference in the function of the gene. But clinically, we also looked at patients who have a high expression of the gene and what their survival outcome was. So this is panel G shown here, and if we look at the difference in survival between patients who have high expression and are African American or who have high expression and are European, we find that African Americans who express this gene have a lower probability of survival, as opposed to African Americans who have, for instance, low expression of the gene. So here we see that patients who are African American who have low expression have a similar survival as white patients who have a high expression. And so as we move forward, then we really want to find out then, well, what is this gene? What does it do? It happens to have a domain for enzymatic activity with DNA, somewhat to BRCA. But functionally, what this gene does specifically is resolve chromatin bridges. So BRCA's role is to resolve homologous recombination so that sort of that resolution of the chromatins happens and they can separate in cell division. And then similarly here, if some of that is not resolved, then what ANCL1 does as the cell starts to divide, and then there's this sort of string, if you will, of chromatin that's in between the cells, it actually stops cell division and then meticulously teases this apart so that there are no errors in the DNA, right? If ANCL1 is functional. So ANCL1 is also a LIM3 ortholog. In worms, it's called LIM3. But if you don't have that function of ANCA1, instead what you have is another mechanism of double-strand break repair that causes genome instability. Okay, so it's not sort of this meticulous repair, it's just chopping it and there's a double-strand break. We would expect then that if you don't have the ANCA1 function, similar to if you don't have the BRCA function, then we may see the accumulation of mutations that indicate that loss of function, right? And so that's what we're looking for. If we look at the mutational patterns that are in the tumors of patients who have either the G allele, a combination of them, the T and the G, or they only have the T allele, which is the allele that's primarily in people who are African American, and we look across all of the 30 different types of mutation signatures that they can have, we see a different pattern of mutation signatures happening in individuals who have the T allele. And so what does that actually tell us? So first of all, signature 3 shown here in this Kelly green is representative of the BRCA mutation. So typically if you have a BRCA loss of function or pathogenic BRCA variant, you will see the accumulation of signature 3. But these individuals who have this T allele ankle also now are accumulating a lot more signature 16, signature 5, right? And so what we want to understand is whether or not signature 16 and signature 5 are basically the consequence of not being able to resolve those chromatin bridges and the genomic instability related to that. But as of today, ANCA1 is not even a cancer gene, right? It's not on any mutation. And so we want to try to fix that. I want to change gears a little bit and talk about gene expression, because gene expression is really what determines a tumor phenotype, right? It's the network of genes and how they interact. And the first thing that we do is measure genetic ancestry. And one of the things I wanted to point out about this paper is that when we looked across each patient, in this figure, every patient is a bar on the graph. At the top, that panel is representative of the global ancestry, so at a global level. So East Asian is represented in red, South Asian is in blues, European is in green, Native American is in purple, and then African is in this golden orange. So patients who self-identify as African American, of course, have this range of genetic ancestry, which we expected to see. And then if we look more carefully at their regional ancestry, which is shown in the bottom panel. So these are regions of Africa represented here, because Africa is a continent, a very large continent. We find that every region of Africa is in the background of African American. So each individual African American has genetic ancestry from every region of Africa, at least the regions that we currently are able to discern. And that means a lot. And it also corresponds with what we know to be the social history of African Americans in this country, as opposed to, for instance, the people who are West African ancestry and current Ghanaian patients is primarily simply West African populations. So just also anecdotally, I'll mention that the Ethiopian population had much more European ancestry than we anticipated, so much more that the sequencing center was alarmed and thought we had contaminated the sample somehow. But anthropologically, if we look at the social history of Ethiopians, we actually know that they shared a border with Eritrea, which was an Italian colony. And the majority of their European ancestry, as you can see by the solid coloring, was Italian. So their DNA in their tumors reveals some of their social history. Now, before we have the ability to measure ancestry, we used self-reported race to define our populations and do comparative studies, for triple negative tumors in the gene expression. And it worked. Like, we do see clearly some differences between this group represented with the red bar and the group represented with the blue bar. Blue bar. And so the blue to green transition here for each of the genes, which are the rows, represents whether the gene is at high expression or low expression. So green represents high, blue represents low. So you clearly see that we can separate the population even by Black and white patients based on this particular set of genes. But when we stripped away race and only used genetic ancestry, the clarity became so much better resolved. The number of genes goes down, but you clearly see a better distinction in terms of the gene expression differences between these two populations, because it's based on their ancestry and not on their race. And it was so well defined that even the European Americans here separated into two separate populations based on the admixture, based on the admixture in their genetic background. So this tells us that utilizing measured ancestry does help us resolve expression regulation across gene networks. And so what we found in the cancer discovery paper, which was the first time African ancestry has been shown to be biologically relevant in triple negative phenotypes, is a host of things. First of all, we found genes that were associated with both African and European ancestry, but we only focused on the African ancestry genes because we've been focused on European genes long enough. We know what's in that book. What we don't know is what's in the African book. And so what we identified were some not necessarily novel pathways, but unpredicted or unconventional activity in those pathways. One of them was the activation of the P53 pathway, which we recently went on to show is a real thing in cells of people who are of Ghanaian descent. I don't have time to show that today. But the outcome of our previous work and the current work that we're doing is really trying to improve the precision of precision medicine, how accurate it can be, and to make the proof of principle that we've been missing a lot of information that could potentially help save the lives of individuals with cancer if we simply broaden our scope to be more inclusive of the diversity in the global population. And so we put forward this table where we show these African specific gene upregulated genes that were somehow associated with African ancestry. At that time, we had not honed in on why, right? So could it be a different transcription factor regulation? It could be epigenetic signatures related to structural changes, but we just wanted to, at that time, just put forward the fact that we could reproduce these gene networks, and we identified a list of them that already had drugs that have been tested in cancer, but were not currently breast cancer options for people with triple negative breast cancer. We've gone on to do CRISPR-Cas9 screens to verify a few of these, including afitinib as a potential drug treatment for triple negative breast cancer, but I don't have time to tell you about that today either. And then the activation of the P53 pathway also revealed that patients could potentially be eligible for things like PARP inhibitor therapy, which we've also gone on to show in cell line studies. But I want to come back to this other bubble that's at the bottom here. So remember I told you before we used just ancestry, we also used self-reported race. So we did both. And what we found was that there is a subset of genes that are strongly associated with race, but have nothing to do with ancestry. So we were curious about what those genes are because they were in our first study, right? But in this study, we kind of filtered them out. And remember, well, maybe I didn't tell you this, but I'll tell you now. We didn't use any Europeans in this analysis. So it wasn't just simply African American versus white, right? We're talking about people who have a shared genetic background of African ancestry, but that the African Americans particularly had a very distinct signature compared to the African patients. And then we even see some resolution between East and West African patients, to be honest. But what we were really interested in was a subset of genes that appear to be upregulated only in the African American population. And when we looked at what those gene networks were, we saw enrichment of pathways related to things like diabetes, obesity, inflammation in adipose tissue, cardiac dysfunction, and all of this, remember, is from tissue from the tumors. So that means that the comorbidities that are in higher prevalence in these patients is actually imprinted on the biology of their tumors. And this is something that has not been characterized. And it's something that clinically hasn't been something that we integrate in our thinking of decision-making for how we treat patients, but these are the patients that are coming into the clinic. So I've just introduced a couple of things to you. I've introduced the fact that we have a genetic, underpinning that's different. There are some gene alleles that are functionally different that could potentially be altering the way our DNA is mutated in that pathway to malignancy that we don't pay attention to. I've also introduced to you that there are cardiac toxicity alleles that could potentially be in higher prevalence in this population that we don't pay attention to. And now I'm telling you that the comorbidities that are in higher prevalence in this population create a very distinct pattern of gene expression and then therefore biology in these tumors, and we don't consider them in clinic. Now, one of the genes that I study more so than others, my favorite gene is called DARK or ACKR1. There's a mutation in the gene that's called Duffy null, and Duffy null is also a blood group. Okay, so that mutation was fixed in Africa, sub-Saharan Africa specifically, because it also confers an immunity to certain forms of malaria. So normally it would be expressed on red blood cells, but being Duffy null means you no longer have expression of the protein on your red blood cells, which is why it's a blood group marker. Now that protein is also the docking site for Plasmodium vivax, so removing it removes the ability of the parasite to cause malaria, right? But what we also know is that the normal function of the gene on red blood cells is that while the red blood cells are moving in circulation, they're also accumulating and sort of sequestering inflammatory chemokines because it's an inflammatory chemokine receptor. ACKR stands for atypical chemokine receptor. And so what we note here is that individuals who are Duffy null, who are going to be mostly African ancestry by virtue of the allele being African specific, because it was fixed in that population. And we know now that it's distributed across the Americas and the Caribbean based on the transatlantic slave trade. So we know, for instance, that African Americans have a 60 to 70% likelihood of being Duffy null. And it's actually something that's tested in blood banks, but it's not necessarily something that's on your donor card, right? But before you have a transplant, if you're of African ancestry, they are going to type you for this allele, and they will have to try to match you with someone who has the same blood group as you. What we found was that the patients who were Duffy null also had a higher amount of inflammatory chemokines in circulation. And what we believe is happening then is even before the malignancy, even before an immunological response to the tumor, that immune cells have been altered in a way that makes the tumors more aggressive and the immune system less effective. We call it immune cell exhaustion. And so a couple of these have already been shown sort of in earlier studies, 15, 20 years ago, in terms of the chemokines that the protein binds to in binding studies. And so what we found was that the prevalence of the chronic inflammation for those particular chemokines were the exact ones that DARK or ACKR1 would normally bind to. And it just so happens that a few of these have also been shown to be exhaustion culprits, if you will. IL-1 beta, for instance, deactivate cytotoxic T cells. And we believe that this is what's causing an adverse immunological phenotype in these tumors. But what's interesting about DARK and Duffy null is that in mouse models, being Duffy null has also been attributed to adiposity in a high-fat diet. And we also see inflammation of that adipose tissue, particularly in DARK knockout mice. And this is also correlated with insulin resistance. And I don't have time to necessarily tell you about each one of these different experiments. I'm sorry, I must have left off. I can send you the reference, but this is a paper that was published about 15 years ago describing the Duffy knockout. And so what we have then is a set of patients who are Duffy null, who actually cannot regulate inflammatory chemokines, but live in an environment that is often triggering inflammation in many different ways. We have an altered immune response that then makes tumors refractory to treatment. But we also have the potential to have epigenetic responses to where we live. So minoritized populations are often marginalized in terms of where they can live, which means in a city, they're going to be in the more industrial areas with more exposure to pollution, less food security. All of that can imprint on the tumor and create distinct biological issues, including pro-inflammatory diets, which is linked to food security. And if you're dumpy null, that means you're going to accumulate this inflammation and it's going to be chronically in your system, creating, for instance, this inflammatory adiposity. So step in, stand by. We know that all of these problems are intersected, that they interact with each other. And so if we're really going to solve the problem of disparities, then we have to capture this information from not only a global perspective, but from an interdisciplinary perspective. And so our team is made up of leaders in the fields of social and societal determinants, exposures, where we do exposomics, genomics, and epigenomics, and then tumor immunology and host immune biology. And then we follow up with our fifth working group or work package, which is our patient involvement team, where these are patient survivors of cancer. We're focused on breast, prostate, and pancreatic. And they will be sort of our ambassadors to the community, right? So before any clinician or scientist asks someone to be a part of the study, the patient involvement team will go and do a needs assessment in the communities where the patients are, and then bring that information back to us. And then we make sure that we focus on those needs. So for instance, if it's air pollution or coal mining, for instance, in Coal City in Nigeria, or here in North Georgia, carpet mills or farmers and pesticides. So we'll be looking specifically for those interactions with the epigenomics and the tumor biology. And then my last slide, might not be my last slide, but I am gonna stop here, is our genomics part. And this is close to my heart because not only am I a genomicist, but I'm the director of the Institute for Translational Genomics. And so one of the things that we plan to do more carefully in a way that's never been done before in this context is create our own human genome references that are specific to the populations that we are studying. And this is for two reasons. Number one, the current human genome reference that we use, that everybody uses, right, has gaps in it. We don't know how big the gaps are until we have the technology that sequences across them. And what we found, and I'll come back to that, is that there's a lot of information in that gap. But we'll also be doing sort of this multidimensional assessment of societal pressures. So not just where a person lives, but how they live and how the way they've lived has impacted them. And then we'll measure all of this and quantify it to determine if we can make sense of, if you will, those interactions. But the pan-genome references that we will create are also going to enlighten us to some of the genetic background differences that just exist across the population. That could also be the reason why a lot of genetic tools don't perform as well in these populations. And one of these, and I think this is my last slide, is we took some of those gaps, right? So there was recently a paper published where the investigators took patients from Nigeria, and these weren't even cancer patients, I believe they were asthma patients. And they sequenced from telomere to telomere, so there are no gaps. And then they aligned it to the human reference and identified what the gaps were. So we took that new genome sequence and we asked the question, number one, are there regulatory regions in this newfound DNA? And yes, we found 11,000 CPG islands that have never been interrogated before. But also, interestingly, we took what we call unmapped reads from our RNA-seq. So every time you do an RNA-seq experiment, you kind of blast it to align it to the human reference. There will be a bucket of them that don't align. And sometimes we think that's just contamination from microbes, chicken, depending on like how we collect the sample, if it's saliva. But in this case, we took the bucket of unmapped reads and we aligned it to the novel context, and lo and behold, about 30% of them actually align to the new regions of the genome. So that means that there are genes in there that are being expressed that, again, we've never been able to interrogate. So as part of Team Standby, what we'll be doing is integrating all this information, looking at these genomic signatures and even down to the single cell level and the state of the cells, using the highest grade technology, state of the art that we can. And the whole premise here is to identify the constellation of factors that really drives, for instance, the disparity at a population level. And I'll stop there, thanking all of my lab members and my collaborators across the world and our funders, and hopefully we have a few minutes for questions. Thank you for your attention. Wow, there are a lot more people online than I thought. Everybody's online. Great, okay, so do I operate the questions or
That's me.
Dr. Akadobi, are you
That's
gonna
fine.
moderate?
Yeah, you want to go ahead, Becky, did you have something?
Yeah, if nobody else, I think Arnetta had her hand up before me. Arnetta?
Arnetta, you can go ahead.
You are on mute, Arnetta, if you're. Yeah, maybe you can go ahead, Ben.
Okay, very nice presentation, Melissa. It's kind of, I was very intrigued with your last slide on the Sambai study. You're recruiting 12,000 cases and 28,000 controls. And you're doing whole genome sequencing. So in what populations are you recruiting from?
Great question. So that's another slide. So we currently have 5 different countries that we're recruiting from in Africa, which include Nigeria, Ghana, South Africa, Ethiopia, Kenya. Kenya might actually be replaced with another East African country. And then we're also recruiting in the United States and in the UK. And in the UK, we're recruiting specifically in London, Scotland, and around Nottingham, because that's where our investigators are located. But we're using the National Health Systems network there in the UK to recruit from that population. Black British primarily, and African Americans primarily. Only about 10% will be of European ancestry.
Yeah, and the reason for my question is really I'm interested in your control group, which is 28,000 people. And again, my questions would relate to how this infrastructure can be leveraged for cardiometabolic diseases. So whether, if there was a possibility of recruiting cases of diabetes, particularly across diabetes phenotypes, that would be a separate kind of a supplementary grant. We could use your control group for the comparison. That is where I was thinking about, because you have all the other measurements. What you need is the phenotype of diabetes.
Yeah, so we're not excluding patients because of comorbidities, because we want to have a real sort of sampling of the true population, especially knowing that diabetes or obesity actually imprint something unique in the tumors. And I'll have to go back and recheck the numbers, but I think our controls are more like 13,000 and the cases are more like 28,000, but it's 40,000 participants total. But yes, absolutely, we plan on having some spinoffs. Actually, I have to try and hold them back. So most of the investigators that are on the team came in the room with, spinoffs. And so they've already started, trying to write grants. But we will also have a mechanism for collaborators to join and tell us, if there's something we can help leverage in our study, and if there is an opportunity to expand the study, to pay closer attention to details such as comorbidities. So there are people who are interested in obesity and adiposity deposition, and they'll be looking at that. And then we also have an interest in, for instance, other infectious diseases. So we know there's a proportion of individuals who, in the past, in our international studies, have a positive HIV diagnosis. We, in the past, haven't had enough of those patients to do a separate study of the impact of HIV on the tumor biology, but more broadly, infectious history is, I think, going to be really important, particularly for people who are definitely null, because that'll help us understand sort of the timeline to accumulate a chronic inflammatory state. So yes.
Just one question about your triple negative breast cancer. I mean, I'm just curious, is there a animal model for it? And my more important question is, if you're promoting screening for triple negative breast cancer, is there a danger that it could lead to unnecessary mastectomies?
Yeah, so that, I'll answer your last question first. That has often been sort of the argument for not increasing, screening by lowering the age, because we know actually triple negative is an earlier onset disease, and oftentimes we'll see a significant portion of African American women being diagnosed earlier than what is the age at screening in terms of screening guidelines. But the pushback for that has always been over treatment, over biopsying, because these women also have more dense breast tissue, for instance. I don't know if I answered your question, but I will say that other modalities of screening are becoming much more favorable. So liquid biopsies, for instance, if those ever actually get worked out to become a screening tool, we know that we can use them for monitoring cancers once they've been diagnosed. But if it's possible to also use that as an option to identify a tumor or a patient with cancer, then I think we would be able to turn the tide there, where we're not necessarily, over treating, if you will, with invasive measures. To answer your first question about the mouse model, so we have a mouse model that 100% penetrance of breast cancer, they happen to be triple negative, right? But we don't actually know what the mechanism is that drives triple negative breast cancer. We just know the snapshot after the tumor is formed is that those hormone receptors are not present, but we don't necessarily have a full appreciation for what drives that phenotype. There are some schools of thought that they were always positive and turn negative, right? And then there are schools of thought that it's a completely different etiology, right? That it's some other growth factor or something environmental related to, for instance, inflammation, because it shares some traits, if you will, with inflammatory breast cancer. So to answer your question, there are ways you can generate a mouse that has triple negative breast cancer, but we don't necessarily have a genetic model that is the model, right, that represents TNBC. And largely also because there are about 5 different forms of triple negative breast cancer. So remember, TNBC is a diagnosis of exclusion. So within that bucket, there's at least 5 other types of breast cancer that can be defined by other markers.
It is 1 o'clock. Those of you, thank you for coming. Can you take one more question, Melissa?
Yeah, sure.
Okay, Vincent Marconi has a question. Go ahead and unmute yourself.
Yeah, hi, Melissa. That was a fantastic talk. Really enjoyed it. I've been sort of doing similar kind of work in the HIV space. We actually studied years ago the role of Duffy antigen for HIV susceptibility and disease progression. And I guess I'm wondering if in the breast cancer and cancer literature, you might be in a closer space to where we're hoping to get in the HIV world, whereby the convenience of using a race or ethnicity self-report is helpful for being able to stratify and adjust and provide explanation for differences in disease outcome. But I'm just wondering, are you at a place or getting closer to a place where you could use, area deprivation indices, individual levels of socioeconomic deprivation, and other markers and geography and other things along those lines, along with Ancestry, that you could eventually foresee or envisage not needing those self-reported markers?
Yeah, absolutely. So to your point, right, the neighborhood deprivation index, actually the lead for that work package is Laura McCullough, who's a faculty member here. And a lot of her work actually shows that the current formula, if you will, of neighborhood deprivation indices don't perform the same as anything else that's been, created to date, don't perform the same in Black women, particularly Black women who are at higher SDS status. I think in her work, she says that at the highest SES status, the gap of mortality is the highest as well. It's 126% more likely that a woman with breast cancer will have an adverse outcome compared to white women in the highest strata. So that means it's not necessarily neighborhood area deprivation, at least the way that we define it, by things that are related to socioeconomic status, right? So we're currently, neighborhood deprivation is defined by the conveniences around you, the income, the education around you, but it doesn't necessarily attribute other forms of stress, social isolation that may come at that higher SES status. So yes, we plan on using some marker that helps us stratify the factors that are in the environment or in the lived experience of these patients, but it's probably not going to be what is currently used, right? So that's part of what our team is going to do, is create better best practices for that. And then I guess the other part to your question is, instead of self-identity, I don't know. I think that for the genomic side, definitely, because I believe we're just at a moment in history. I say this all the time. We're just in the moment of history where we're making the point that there are genetic differences that have not been identified and have not been studied across the broader population, whether it's of African, Asian, Middle Eastern, Native American, South American. There's just so much more rich genetic information that we have not identified and utilized in our research that could potentially be beneficial in terms of identifying what the mechanisms are that drive cancer.